Microscopic autoradiography is a powerful technique in cellular and molecular biology. It allows scientists to visualize the location of radioactive substances within cells and tissues. By using a radioactive version of the building blocks of a protein, we can track where they end up. This signal is captured on a special photographic film, creating a map of the protein's location. This technique has been instrumental in understanding various biological processes. It allows researchers to study DNA replication, RNA synthesis, and protein localization. The use of autoradiography has revolutionized our understanding of cellular processes. This knowledge is crucial for developing new drugs, understanding diseases, and advancing our knowledge of life itself. Microscopic autoradiography involves several key steps. The first step is incorporating radioactive isotopes, which act as tracers. Common isotopes used include tritium, carbon-14, and phosphorus-32. Next, the biological specimen, such as a tissue section or cell culture, is prepared. The specimen is then coated with a photographic emulsion containing silver halide crystals. The coated specimen is exposed in a light-proof container for a specific duration. During this exposure, the radiation from the isotopes interacts with the silver halide crystals. This interaction creates a latent image, which is invisible to the naked eye. Finally, the exposed emulsion is developed, converting the latent image into a visible one. The areas where the radioactive isotopes were present appear as dark spots or grains. These spots provide a visual representation of the distribution of the radioactive substance within the specimen. The process of autoradiography involves meticulous preparation and execution. Firstly, the sample is fixed using chemicals like formaldehyde to preserve its structure. Then, it's embedded in a medium like paraffin wax to provide support during sectioning. The sample is then sliced into thin sections using a microtome. These thin sections are mounted on glass slides, ready for the next steps. Coating the prepared sample with a photographic emulsion is critical. This emulsion contains silver halide crystals sensitive to radiation. The emulsion can be applied using either the dipping or the stripping film method. Once coated, the slides are left to dry in a dust-free environment. After drying, the slides are placed in a light-proof box for exposure. The duration of exposure depends on the type and amount of radioactive isotope used. Following exposure, the slides are developed using photographic chemicals. The developed slides are then analyzed under a microscope. The distribution and density of these grains provide valuable information about the localization and quantity of the labeled substance within the sample. Autoradiography has proven to be an invaluable tool in various scientific disciplines. In cellular and molecular biology, it is used to study various cellular processes. It allows researchers to track the movement of molecules within cells and investigate gene expression patterns. In drug discovery, autoradiography helps study drug distribution and target engagement. By radiolabeling drug candidates, scientists can track their movement and accumulation within tissues. In environmental science, autoradiography tracks the fate and transport of pollutants. By labeling pollutants with radioactive isotopes, researchers can assess their impact on ecosystems. In medical diagnosis, it can identify the presence and location of tumors. In neuroscience, autoradiography maps the distribution of neurotransmitters and receptors in the brain. Like any scientific technique, autoradiography comes with its own set of advantages and limitations. First, high sensitivity. Autoradiography is highly sensitive, allowing the detection of even minute amounts of radioactive substances. This sensitivity makes it ideal for studying processes involving low concentrations of molecules. Second, spatial resolution. This technique offers excellent spatial resolution, enabling researchers to pinpoint the precise location of radioactive substances within cells and tissues. Third, versatility. Autoradiography can be applied to a wide range of biological samples, including tissue sections, cell cultures, and even whole organisms. Now let's look at the limitations. Handling radioactive materials requires strict safety protocols and specialized facilities. Autoradiography can be time-consuming, especially with long exposure times. Despite its limitations, autoradiography remains a valuable tool in scientific research, with high sensitivity and spatial resolution, making it indispensable for studying a wide range of biological processes.